Dear audience, it is a pleasure to speak to you about my work. My purpose today is to show you possibilities for analyzing the genomes in public databases and genomes you deciphered by yourself when looking for exciting proteins for vaccines and diagnostics in bacteria. As an introduction to the theme, I would like to say that there is excellent software and databases, just citing three of them, UniProt, Gene Ontology, and StringDB, allowing us to inspect our automatically annotated genomes under the perspective of probable function, molecular process, and subcellular localization. We can think about these three features as three-dimensional axes of study universally known from, for instance, the Gene Ontology database. Despite the importance of understanding these three features for proteins under investigation, we also learned they are not enough to elucidate bacterial infection mechanisms. We cannot capture the interplay among proteins using just these three features. A method to capture such interplay could be creating a fourth dimension in our investigation, the interaction dimension. In 2016, we tried to obtain the interaction dimension using the three features documented in Gene Ontology. Our idea was to annotate an interaction for each pair of proteins possessing similar characteristics, having comparable functions, occurring in related molecular processes, and present in identical subcellular location. The article using the Gene Ontology tool to produce de novo protein-protein interaction networks with ISA relationship produced a negative result only with these features. However, when we added the part conserved neighborhood using the protein suffix numbers, the results were less disappointing. After that, we decided that if we would like to count on reliable ab initio prediction for protein interaction, we must build our software. Another possibility for relying on trustable interaction protein prediction is based on experiments like gene expression profiling. The rationale is that proteins expressed together are prone to interact in a molecular process. In doing so, we also incorporate the fourth dimension into our investigation by annotating each pair of proteins expressed together. For instance, if 10 proteins are highly expressed in an experiment, all interact among themselves. We can create 10 turns 9, resulting in 90 interaction patterns in our genome under analysis. The main drawback here is that gene expression profiling experiments designed by microarrays or RNA sequencing are expensive and demand a complex laboratory structure for researchers outside of developed countries. However, we believe there are other forms to infer interaction without a wet laboratory. Genomes conserve evolutionary features like conserved neighborhoods, phylogenetic profiles, and gene fusions. Meanwhile, I turned my attention to exploring interaction networks publicly available. Software like StringDB.org offers interaction networks for thousands of organisms, exploring since from software-predicted genomic features to experimental evidence of protein interactions. What could we get by analyzing these webs? What methodology could we use to analyze these graphs? In our research, we figure out graph statistics, also called centrality measures, which are valid and accepted by the general scientific community. The most general statistic looks to be the degree or degree centrality, referred to as hubness. The idea is the more central a node in a graph, the more connections it has. However, hubness does not look like a promising statistic for target selection because essential proteins, for instance, are not good candidates for a knockout experiment. Another well-known statistic is the Betweenness Centrality, which highlights nodes in a graph traversed in most, considering all pairs of nodes and all paths connecting any pair. Betweenness centrality is also a method to contrast essential proteins in a graph, not by immediately connected vertices but indirectly. In opposition, bridging centrality does not focus on crucial proteins. Bridging centrality is the product of two others, betweenness centrality and bridging coefficient. The last one is an inverse of degree centrality. The consequence is a metric highlighting node relevant to the general graph traverse but possessing few directed, connected vertices. Proteins highlighted by this statistic have a bias to be not essential and connect clusters of proteins. It is possible, for instance, for the inactivation of proteins with higher values of bridging centrality also knock out an infectious process caused by a pathogen. At the time of our research, no software allows us to calculate this statistic in an interaction network. In 2021, we published the article A Bridging Centrality Plugin for Genpe and a case study for Mycobacterium tuberculosis H37RV to allow the scientific community to study the publicly available genomes of any organism under the light of non-essential proteins connecting clusters of proteins. In our article, we illustrated a hypothesis of a relationship among proteins previously unrelated. We based our assumptions on a four-dimensional scenario, the three features documented by gene ontology, and the bridging centrality inferred by an interaction network. StringDB.org uses conserved neighborhoods, phylogenetic profiles, and gene fusions features, plus others, including literature evidence of interactions. Today it has about 14,000 genomes. However, the number of novel in-house genomes we can produce nowadays could overwhelm this magnitude of genomes with interaction networks available in about 1 year. The difference between the GenBank releases 249, April 2022, and 248, February 2022, is 1,182, and 34 sequences, excluding WGS sequences. Yet, our in-house sequencing genomes are not available in public databases. Even after publishing a novel genome sequencing, it takes time until a public database, like StringDB.org, incorporates it as part of its pipeline. To solve these difficulties, in 2021, we also published Genpe, standalone software for creating protein interaction networks from genomes. Genpe is like the ab initio generation of protein-protein interactions or from genes to protein-protein interactions. The fundamental evolutionary features explored by Genpe are conserved neighborhoods and phylogenetic profiles, but gene fusions are also possible. It takes any set of genomes, represented by their proteins ordered like in a chromosome. It creates an interaction network for each genome from scratch, included in one analysis round. Despite using two evolutionary features, we showed that using less than 0.009% of the number of genomes present in StringDB.org or 50 genomes, we could map 46% of the web predicted by StringDB.org. On the other hand, StringDB.org predicted only 14% of our net. We also showed that our interaction networks are trustable because we used them to infer phylogenetical analysis of the genus Corynebacterium. Our phylogenetical analysis allows us to separate the two biovars of Corynebacterium pseudotuberculosis species correctly. We should warn a future Genpe user, there is no closed formula to obtain a reliable interaction network for a set of genomes. We can trigger several rounds of execution until we get a reliable network. Some rules of thumb are the number of proteins present in the genome of interest, the average degree of the nodes, the graph density, and the degree distribution or the probability density function for the degree distribution. We expect the number of proteins close to the genome's total, an average degree at a couple of hundreds, a graph density close to 10%, and degree distribution following a power law distribution, many nodes with small degrees, few nodes with high degrees. However, even in StringDB.org, graphs infringe these rules of thumb, with about 1,000 vertices as the average degree, for example. To illustrate what was said, let us show images of the software. The first out of 4 images show some Linux commands and their outcomes. However, Genpe also works on the Microsoft Windows platform. From left to right, up to down, we list the directory content with files and folders created by the Genpe software. A grep command counts how many protein sequences there are in each file. A cat command lists the summary report of the Genpe execution, depicting the number of interactions identified by the conserved neighborhood and phylogenetic profile features by genomes. The right panel shows us the content of the first protein present in each genome analyzed. This is what Genpe expects to proceed with its analysis. The second of 4 pictures show us a Genpe screen of one of the networks created by Genpe. We added additional data concerning subcellular localization from an external tool. Most proteins were classified as cytoplasmatic, followed by a few potentially exposed in the surface, membrane, and secreted ones. The proteins are the nodes, and the vertices are their interactions annotated by Genpe. Below the mouse cursor, we can see the Bridging Centrality plugin installed and ready to be called in Genpe. Indeed, we already ran it. Following, we show the third of 4 pictures. The third figure is a pretty print of the previous image. Because we already have bridging centrality calculated, we highlighted the top scored proteins with larger nodes. In this run, Genpe found 172 proteins conserved out of 621, most of which were cytoplasmatic. For the organism Buchnera aphidicola, we expect this result since this organism is a symbiont. Because of the symbiosis, most conserved proteins are the essential ones, cytoplasmatic ones, and have the highest bridging centrality scores. Our final picture came from our bridging centrality article. We downloaded the interaction network for the Mycobacterium tuberculosis H37RV and filtered that for the higher probabilities interactions. We formatted the resulting network like our previous image, including protein localization. Moreover, we created a database containing several annotation features for the genome. This data allowed us to infer a relationship between proteins RV3779 and RV3781, previously unrelated. In our hypothesis, the protein RV3779, part of the glycosyltransferase cluster, could act as a shipping channel for the T7SS exportation system to communicate to the outer layer of Mycobacterium tuberculosis H37RV. In conclusion, we are proud to deliver to the scientific community software to grant the analysis of an interesting statistic as bridging centrality. This metric can help researchers on mapping target proteins for fighting pathogenic bacteria. In case of the inexistence of an interaction network for a specific organism, we offer the software Genpe to create from scratch a reliable protein network based on evolutionary features. The Bridging Centrality plugin for Genpe and Genpe are both open-source software. Thank you for your valuable time. Regards, Anderson Santos.